Welcome to this virtual tour of the Scientific Computing Research Unit. I'm Kevin Naidu, the unit's director, and I'll be leading you through this virtual walk. The SCRU, or SCRU, is an interdisciplinary unit active across three faculties: science, engineering, and health. Inspired by a self-imposed mandate of leading innovative scientific solutions through the rigor of computational models and the observational insights gained from data analytics. The core members are based in the academic departments of chemistry and electrical engineering, and there is close collaboration with academic departments in health sciences. While we have different individual research interests, the overlapping research space we share in SCRU is for the development of the computational methods that affect diseases, and more recently for the experimental testing of the accuracy and effectiveness of the computational models we produce. The students and postdoctoral fellows we train have basic BSc Honours, BSc Engineering, and advanced MSc and PhD degrees in applied mathematics, chemistry, chemical and electrical engineering, molecular biology, and physics. Through this virtual tour, you'll find our mantra, "Science through Computing," has been a guide to our research. And this has taken the computer board to the laboratory bench to the patient bedside. Let's begin. The focus of the Computational and Modeling Laboratory is the development of algorithms, modules, and packages needed for life sciences modeling. The SCRU hardware platform includes state-of-the-art GPU clusters, data servers, and InfiniBand clusters. The compute capability is modest and designed for co-development, model testing, and short simulations prior to using either the national facility, the CHPC, or the UCT and regional facility, ILIFU. SCRU developed free energy, force-induced, that is phi, coarse-grained molecular dynamics is used to generate the protein structures. These structures are used to develop models for reaction simulations. Following the structure modeling process, the multidimensional reaction dynamics of complex glycoenzymes catalytic mechanisms are made possible from the SCRU's development of the free energy from adaptive reaction coordinate forces, that's the FESCF library. Ab initio quantum dynamics and free energy methods make possible the accurate depiction of glycoenzyme reactions. The ab initio quantum and hybrid QMMM simulations are uniquely possible using the SCRU-developed quantum supercharger library, that is the QSL. The QSL is capable of rapidly accelerating legacy code such as Games UK and NWChem. The Computation and Modeling Laboratory's primary objective is to use these accurate reaction mechanisms to design therapeutics for cancer and respiratory diseases. The designed therapeutics are synthesized and tested in the Biomedical Laboratory. In addition to these objectives, the complementary groups in computational chemistry, one of them led by Christopher Barnett, aims to discover and understand molecular recognition events and to assist in the prediction and decision-making in drug discovery. The group for ionic liquid development, led by Gerard Venter, aims to develop ionic liquids as possible alternatives to glycoenzymes. In the Informatics and Visualization Laboratory, analytics and visualization tools are designed and implemented to infer decisions from large heterogeneous data sets, the so-called big data. The Visualization Studio is a key facility that provides an environment where researchers can visually immerse themselves in high-resolution simulation data and large informatics datasets. A large visualization and video wall, the first one installed at UCT, provides the visual immersion environment, giving researchers the ability to compare analytics models and visualize complex molecular structures. Touch and 3D navigation devices allow users to interactively explore and manipulate complex data. Visual workflows analysis and visual summaries of analytics data make it easy for novice users to make rapid progress in their projects. The Machine Learning and Bioinformatics group developed machine learning techniques and informatics platforms. Two notable outputs here are, firstly, the denoising autoencoder self-organizing map, that is DASIM. This is a flagship project of the SCRU. DASIM is a state-of-the-art unsupervised machine learning method that has been published in the leading journal Neural Networks. It can discover cancer classes and subclasses from noisy biological data. Our clinical studies rely on this non-linear method using gene features derived from our genomics patents. The preliminary results promise a clinically useful gene expression biomarker. Another impact output is the Biomolecular Reaction Interaction Dynamics Global Environment Bridge. The Bridge platform is an open source and open science project that enables UCT and South African, African and international life scientists to connect genomics data to model targeted protein structures and to ultimately undertake state-of-the-art drug design. The Glycobiomedical Science Laboratory has two central themes: understanding the role of glycoenzymes in cancer and respiratory diseases. Both themes are tackled from the premise that systems of glycoenzyme reactions enable biological pathways that initiate and progress disease. The experimental methods range from glycoenzyme protein expression, inhibitor synthesis, through to kinetics and inhibitor high-throughput screening assays. All experiments are informed by our computational systems biology discoveries of glycoenzyme networks and our computational reaction dynamics modeling of key enzymes in those networks. Inhibitors designed in the computational laboratory are synthesized by the organic synthesis group and tested by the glycoenzyme high-throughput screening and assay group. Assays are developed and optimized and used to generate data which tests the hypothesis of the computational research. The organic synthesis group, this is work done here, is done in collaboration with Roger Hunter, has a focus on the synthesis of the transition state analog glycomimetics for cellular transferases. These are drug targets for cancer types and subtypes. They also focus on neuramidases that are drug targets for respiratory diseases. The glycoenzyme high-throughput screening and assay group, done in collaboration with Ed Starak and Anwar Jardine, implement as well as develop assays and protein expression methods to analyze catalytic reaction mechanisms that validate the computational models and design. They perform inhibition assays that assist in the lead development of transition state analog glycomimetic ligands in target glycoenzymes. The combination of the four subdivisions allows the Glycobiomedical Science Laboratory to provide a pipeline from plasmid design and enzyme expression through to enzymatic and whole cell testing for the design of inhibitors of strategically importance to the cancer and respiratory disease programs. From the start, the Computing and Modeling Laboratory developed a set of bleeding-edge methods using state-of-the-art software design. These were turned into reliable academic software delivering reliable, rigorous modeling of complex enzymatic processes. Understanding that chemical discoveries rely on biological insights, the informatics laboratory was built, and these were made to discover enzymatic patterns in glycosylation through the development of novel machine learning methods applied to differential gene expression. However, computer models are always treated with suspicion if they are not subjected to experimental testing. Complicating this was the absence of an experimental glycoenzyme specialization in South Africa. The SCRU rose to the challenge and built a glycobiomedical science in-house facility with the capability of expressing glycoenzymes, custom building assays, synthesizing drug hits, and testing them using high-throughput screening methods. All of this was possible through the combined skill sets of talented students trained over several years by SCRU's cohesive and multi-skilled academic staff. Thank you.